Bismillahirrahmanirrahim. Assalamu alaikum dear students. Welcome to your channel The Anatomy Canvas. Today we are going to discuss with the histology of the large intestine and appendix. So functions of the large intestine are to reabsorb water and compact the material into feces, absorb vitamins produced by bacteria, and store the fecal matter prior to the defecation. Areas of the colon are the cecum, the appendix, the ascending, transverse, descending, and sigmoid colon, and then the rectum and the anal canal. So distinctive features at the gross levels are the tinea coli which are present. You can see these are the tinea coli which are present over here. These are the longitudinally spaced muscle band of the muscularis externa. Then the hastrations. So these are the hastrations. You can see these are the hastrations which are temporary folds present. And then the omental appendices. These are the adipose tissue which are present in omentum, and these are called the omental appendices present over. These are the gross features present at the large intestine. So regarding histology of the large intestine, important negative finding is absence of villi and the plica circularis. Then the presence of the goblet cells and number of the goblet cells increases to up to 4:1 in a first part of the large intestine, with the ratio of 4 columnar cells to 1 goblet cells to the ratio of 1:1 at the last part of the large intestine. Also the deep intestinal glands are also present. So mucosa first, as you know, all other tubular part of the GIT, like all other parts, the large intestine also has a 4 layer, that is the mucosa, the submucosa, the muscularis externa, and serosa. So in case of the mucosa is having the large number of the straight tubular gland. Smooth surface, neither plica nor villi, both are absent. Epithelium is simple columnar in nature with enterocytes and the goblet cells. Ratio of the columnar cell to goblet cell is 4:1 in first part of the large intestine, and it increases to up to 1:1 at the end of the large intestine. Another important finding is that the no lymphatic vessels or no lymphatic capillaries are present in core of the lamina propria, and this has a very important significance from pathology point of view, because of the cancers of the large intestine, even they are very aggressive, they involve whole of the epithelium, but because of the no lymphatic vessels in core of the lamina propria, so the metastasize later when they reach up to the lymphatic vessels of the submucosa. So this is a significance when no lymphatic vessels are present. That's why the spread of the cancers of the colon is in late stages. Another important point is the glycocalyx. Like small intestine, glycocalyx is also present in the large intestine. But another important point from your MCQ's point of view is that in the small intestine, glycocalyx contains enzymes which help in digestion. But in case of the large intestine, there are no enzymes present in the glycocalyx. So this is the histological section, H&E stain section of the mucosa of the large intestine. Here you can see the straight surface. There are no villi present over here, neither the plica circularis are formed. The epithelium is a simple columnar in nature with a large number of the goblet cells are present. These are the glands which are deep and straight tubular intestinal glands. In between glands you can see large number of the lamina propria containing high cellularity, and then the muscularis mucosae is present over here. Here you can see a lymphatic nodule is also present. This is the higher magnification of the epithelium. You can appreciate epithelium is simple columnar and having a large number of the goblet cells. Even these goblet cells are present in the replicative zone at the basis of the gland, and from there they start secreting the mucus until they reach the surface where they have been exhausted and they are shed at the surface. So the lifespan of the enterocyte is nearly 6 days, and lifespan of the goblet cell is nearly 4 weeks. Another diagram showing you the mucosa and the submucosa of the large intestine. Here you can see the surface. This is the flat surface. These are the intestinal glands. These are straight tubular glands which reaches up to the level of the muscularis mucosae. Here is the muscularis mucosae and a well developed large lymphatic nodule, which is a secondary nodule which breach the muscularis mucosa and reach up to the level of the submucosa. So this is the submucosa, you can see over here, and this is the large lymphatic nodule which is present. Another important point over here is that the large intestine has a very well developed GALT, that is gut associated lymphatic tissue. So in submucosa you can also see the blood vessels which are present over here, and also the adipose tissue is present. Then is the submucosa, which is the dense irregular connective tissue. Lymphatic vessels in submucosa form a network around the muscularis externi. Then the muscularis externi and serosa. Muscularis externi is the inner circularly arranged muscles and outer longitudinally arranged muscle with three equally spaced bands called the tinea coli. So this is again another beautiful section through the large intestine. Here you can see mucosa and the submucosa, and then the muscularis externa, and this is the inner circularly arranged smooth muscle layer and outer longitudinally arranged smooth muscle layers, and these longitudinally arranged smooth muscle layer forming bands of tinea coli at certain places. Next is the appendix. Appendix lie at the junction of both the small intestine and large intestine, and it has typical four courts. The characteristic features are the thick wall but relatively narrow lumen, which is stellate or irregular in shape. So this is the cross section of the appendix. You can see the whole lumen of the appendix in the microscopy as well, which is another very important identification point for your slide as well. Next you can see that the mucosa is arranged like that of the large intestine, that there are no villi present over here, surface is smooth, and also no plica are present. The intestinal glands are deep and they reach up to the muscularis mucosae. Another important point is large number of lymphatic nodules are present, and large aggregates of the lymphocytes here give it an appearance of like an organ of the lymphoid system. So these lymphatic nodules are so much that they even breach the muscularis mucosa, and they are present in the submucosa in certain even sections of the appendix. You cannot appreciate any difference between the mucosa and the submucosa. Whole of the space will be covered by the lymphatic nodules and lymphocytes. Another important point, as I already have mentioned, that this is present at the junction of both small intestine and large intestine. So the mucosa has arrangement like that of the large intestine, but the cells which are present in the glands resemble that of the small intestine. Outer to mucosa is the muscularis externa, which again consists of the two layers, the inner circularly arranged and outer longitudinally arranged, and outer to that is the serosa. Serosa means it is covered by the peritoneum, connective tissue covered by peritoneum. So this is the whole diagram of the appendix. This is the diagram of the appendix at higher magnification. This is the luminal side which contains some debris over here. This is the epithelium, the simple columnar epithelium, and it resembles the epithelium of the small intestine, but arrangement matches that of the large intestine. These are the lymphatic nodules. This one is the primary nodule, this one is the secondary nodule. Here you can see this is the submucosa, and then the muscularis externa and the serosa. So this area is the rectum, and this is the anorectal junction. This is the transverse rectal fold, and this is the right pararectal fold, and this is the ampulla of the rectum, and here is the junction of the rectum and anal canal. Here you can see the longitudinal columns, the anal columns, and in between the anal columns there are the depressions called the anal sinuses. This one is the internal anal sphincter. So this is the whole more diagram, another important point you must have to remember that the tinea coli are absent in the rectum, and in case of the rectum, the longitudinal muscle, smooth muscle layer is arranged, it is covering whole of the rectum. Also in the lower part of the rectum, the inner circularly arranged smooth muscle layer forms the internal anal sphincter as well. So this is the diagram from the anorectal junction. This is the area of the rectum where you can see the typical epithelium of the large intestine, the flat epithelium, straight surface, and the glands which are present, and then the muscularis mucosae. Here, this is the area where the transition take place. Here you can see the muscularis mucosae abruptly end over here. Also the intestinal glands are absent over here. This area is shown in enlarged magnification over here. Here you can see the muscularis mucosae is ending over here. Also the intestinal glands ends over here. The simple columnar epithelium with goblet cell, which is characteristics of this large intestine, is changes first initially to the stratified cuboidal epithelium, then it becomes the stratified columnar epithelium, later on it becomes the stratified squamous epithelium. So this area is also shown in this magnification. Here the epithelium is a stratified cuboidal. Here the epithelium is a stratified columnar. So this is the area of the transition from the area of the large intestine to the area of the rectum, and this is the internal anal sphincter. This is actually the circularly arranged smooth muscle cells of muscularis externa of the large intestine, that is the rectum, which is now forming the internal anal sphincter, and this is the lymphatic nodule, and in this area you can see epithelium now has become the stratified squamous non-keratinized. This is the magnification from this area. You can appreciate stratified squamous non-keratinized type of epithelium, and this epithelium now changes to stratified squamous keratinized epithelium of the anus properly. And below the epithelium you can see the submucosa as well, containing the glands and the anal plexus of the superior rectal artery, which is forming the internal hemorrhoids. So this is whole story of the rectum, the anorectal junction, and the anal canal. Change of epithelium, change of the characteristics of the mucosa, change of the internal anal sphincter, epithelium now becoming stratified squamous. Here it is stratified columnar, initially it is stratified cuboidal, here it is simple columnar, and here it becomes the stratified squamous keratinized, which at outer area will become the true skin. So this section, this is the section of the external anal sphincter, which is a skeletal muscle and which is derived from the skeletal muscles of the perineum. So this is the diagram of the anal canal and the rectum. And now I complete our today's class. These are the references: the basic histology by Leikusen, histology text and atlas by Jan Quera, and histology text and atlas by the H. Ross. And thank you, our dear students, for your attention. And after that we will continue with the accessory digestive gland in the next class. Till then, Allah Hafiz.